Titration by Hannah Kaplan. Titration is the process of calculating the concentration of a solute in a solution. This is done by using a standard solution, or a solution with a known concentration, and adding the solution until an endpoint is reached. There are a few different ways to tell if the equivalence point is reached. The one we used in class was by measuring the conductivity of the solution to find the equivalence point. As we added the standard solution, the ions in the solution we were titrating were being used in the reaction to form a precipitate. This decreased the conductivity of the solution. When the solution began to increase in conductivity, we knew that all of the ions were used up and the reaction was complete. For acid-base titrations, the pH of the solution can be the indicator of the equivalence point. Possible ways of measuring pH can include a pH electrode or various different chemical indicators. In some reactions, a color change in the solution can indicate that equivalence point is reached. For calculating the concentration of the unknown solution, you can use the volume that we found that was necessary to reach the equivalence point of the volume of the known solution. And then you can use the molarity of that known solution to find the number of moles of solute in the standard solution. And then you can use the stoichiometric ratio to find the moles of solute in the unknown solution. And then using the volume of the unknown solution and the number of moles you calculated, you can find the concentration. Efficiency in implementation. First, it's better to use a gross trial with larger increments to predict where the endpoint will be, and then have other fine trials with smaller increments. And when you're adding the known solution, it's important to use these smaller increments as you near the endpoint, because this reduces the intervals of volume that the endpoint would have occurred between. However, you do not need to do this until you're close to endpoint, because it's unnecessary and will take extra time. And part of the reason why it'll take so much time is because you have to wait for everything to react in the solution. And if you don't, then it would be a source of error if not everything has reacted. For safety, you need to keep the acids and bases away from each other to prevent an unwanted reaction. And when you're diluting an acid, add the acid to the water. Do not add the water to the acid, because that may cause the acid to splash out of the container. Some of the chemicals used can be dangerous, so make sure to wear all the proper attire, lab coat, and goggles. For real-world applications and possible uses, titration can be used to find the concentrations of certain chemicals in the blood and urine. The amount of glucose in the blood for diabetics can be found by using titration, and the presence of HCG in urine to test for pregnancy. Titration can also be used to determine the amount of salt, fat, sugar, and vitamins in food, and it can also be used to determine the acidity of aged wines and cheeses.